So hello friends, good evening. So let's start today's session. So the topic for the day is difference gel electrophoresis called DG and molded off. So today we'll cover 2D DG basics, protein identification using molding, time of flight, and proteomics experiment data analysis and its challenges. So let me introduce myself. My name is Ankit Kumar Malik, a PhD PMRF Fellow from the Department of Pharmaceutical Engineering and Technology, IIT BHU. And the course instructor for the same is Professor Sanjeeva Srivastav from IIT Bombay. Let's start. So first we'll know about what difference gel electrophoresis is. So basically it is pronounced by the name called DG, DIGE. So the two-dimensional DG method was basically aimed to overcome the gel-to-gel variability which was observed two-dimensional difference gel electrophoresis, so called 2D DG. So in the two-dimensional electrophoresis, it was allowed the multiplexing examples. So this method was first reported by Unlu et al. In 1997, and this method finally became very user-friendly. So we'll start from the topic difference gel electrophoresis. So the 2D DG method was basically aimed to overcome the gel-to-gel variability which was observed two-dimensional difference gel electrophoresis called 2D DG. So in the two-dimensional electrophoresis, and it was allowed the multiplexing examples. So this method was first reported by Ongo et al. In 1997, and this method finally became very user-friendly, and now thousands of laboratories worldwide are using this technology to overcome the gel-to-gel variations encountered by traditional or conventional 2D electrophoresis. Okay, so this method, the DG method, uh, this difference gel electrophoresis in layman language called DG. So in the entire lecture, I'll name this term as a DG. Okay, so this involves protein labeling and separation techniques. So the optical detection of the proteins is done by using the fluorescent tags. This method performs the linear detection in the wide range of the protein abundance. So overall, one can expect a very sensitive method for giving to rise to a very large dynamic range of the protein detection by using fluorescent tags, and this method is giving multiplexing and separating proteins on one gel. So it is expected that it will avoid a lot of difficulties which are encountered in the traditional 2D gel. Okay. So we'll go through briefly about the principle involved in DG technology. So we have to discuss how to do the protein labeling, then what is the need for doing the internal standards. So let us talk step by step. As we discussed, this is a commonly used fluorescent protein. So labeling reagents for the DG is the N-hydroxysuccinyldyl or NHS ether derivative of cyanine dyes such as Cy2, C3, Cy3 and Cy5 are used in this method. So the Cy3 and Cy5 are used to label your control and your treatments. So one should always try to dyes swapping so that there is no dyes bias. So if in one sample you are adding the Cy3 in control and Cy5 in the treatment, so in the other sample you should do the vice versa. So like the Cy2 remains consistent for doing the internal standard. Now for Psi3 and Psi5, these dyes, the size and charge is matched so that the labeled samples can co-migrate within the gel. Okay. So now let's talk about the labeling method used in the DG technology. So when we are talking about two-dimensional electrophoresis workflow, so in the last class we had looked at different type of staining method currently available such as Cosme, silver staining, Cipro Ruby, and in that concept I briefly described about cyanine dyes. So now in this technology, the dye G technology, those cyanine dyes are used. So I'll cover the cyanine dyes first. So there are three different cyanine dyes as I mentioned, the Cy3, the Cy5 and Cy2. So the excitation and emission wavelength of these three are defined. So the Site 3 citation wavelength is at 540 nanometers, emission wavelength at 590 nanometers, and Site 5 excitation at 620 nanometers, and emission at 680 nanometers, as you can see in the slide. So the Site 2 excitation at 480 nanometers and emission at 530 nanometers. So these dyes are photostable, so they are pH insensitive and spectrally distinct. By using these dyes, the dye method can be used. Okay. So now let's talk about different type of labeling methods. So in DG there are two different type of labeling methods which are possible. So one is the most commonly used lysine labeling, or other is cysteine labeling. So lysine labeling method, also known as the minimum labeling method, where the charge and size of dyes are matched. So there are no multiple labels, and it is known as minimum labeling because it is only labeling a 3% of all the proteins. So I will give you the reason why that is. We are aiming for very small amount of protein to be labeled, and that will be covered in the next slide. So the saturation labeling is done by using cysteine tagging. So amino acid cysteine is targeted to label, that is charge, neutral and size matched. Multiple labels are used. Okay, so it is very sensitive and used only for Site 3 and Site 5 labeling. So saturation labeling is not too widely used, but this is another method one can use for labeling the proteins, and it can be used in DIG methodology. Okay. So let's talk about the popular of targeting lysine. Okay. So why to label only lysine amino acid? Lysine is preferred amino acid, and as I've shown you in this graph, here the distribution of why lysine labeling. Okay. So amino acid across the proteins and their percentage distribution. So lysine is preferred amino acid to label. Its frequency is greater than 5%. So statistically, almost every protein has at least one lysine residue. So there is a good probability that your protein will have lysine residue and it will get labeled. So if you are targeting the lysine, but if that is not the case, one can always try the saturation labeling of cysteine. Okay. Hmm. So now let's talk in more detail about the minimal labeling of lysine labeling. So the cyanine dyes, they react with the primary amino group of target proteins and the N terminal. Okay. So alpha amino group at the lysine, epsilon amino groups, they are labeled by the process of nucleophilic substitution. So the available lysine labeling would create a very hydrophobic environment of the protein. So that was the case observed by the original paper derived by Unlo et al. As I told you in the first slide. So to avoid that hydrophobic protein atmosphere, that is recommended to do the minimal labeling of only 3% of the whole proteome, so that overall it is not very hydrophobic. Okay. So now in this slide I will describe you the labeling chemistry. So DG labeling chemistry. So as you can see in the structure, here the structure of a cyanine difluor comprised of a positive charged cyanine difluorophore. So a linker and NHS ester reactive group. So at pH 8.5, the NHS ester couples with amino group of lysine in the proteins and it forms the covalent bonds. So the amino group of lysine residue, they are positively charged at neutral or acidic isoelectric points. So dye is also positively charged, so the net pi will be unchanged. So therefore these Psi3 and Psi5, these are not going to alter any mass as well as charge overall, and they are very well spectrally resolved. So people prefer doing lysine labeling. It is just wait a minute. So let's resume the session. So mostly people prefer doing lysine labeling. It is not going to alter overall PI values. It is not going to alter overall mass because both the citrate and sulfide will carry the same mass of these labels. So and there is a good probability that your protein will have lysine residue and it will get labeled because of the cyanine dyes. Okay. So now let's discuss about some advantages and limitations of DG. So there are some advantages like the amount of protein starting material required is very small as compared to the requirement of 2D electrophoresis. So DG is extremely sensitive. So higher reproducibility as two different samples can be analyzed on the same gel. So therefore, differences in protein expression levels are purely attributed to biological variations. So accurate quantitative comparison which can be attributed to the use of internal controls which allows normalization of spot intensities across different gels and increases accuracy of protein expression differences. Okay, so no post electrophoretic processing is necessary and thereby there is a reduction in protein loss particularly in the low molecular weight range. And some of the limitations are like only two different samples can be analyzed on a single gel. For more than two samples, a large number of pairwise comparison would be required. So mass spectrometry based techniques such as called one thing eye track could be used for the 4 or 8 plexing. So spot excision is a problem since spots are not visible to the naked eye and hence required aids such as robotic spot picking. So alternatively, a normal 2D electrophoresis gel has to be run for these samples which is then stained with cosme dye and spots of interest are excised. Okay, and some of like fluorescence detection comes with several inherent problems such as background, the detection of signals from non-protein source, example like dust and residue on plates, so and overlap of signals from different sources. Okay. So now the next topic is moldy. So in this, so let's today let's focus on the moldy time of flight, which is one of the very widely used techniques in proteomics. So this provides a high throughput platform for several applications, including molecular weight determination, protein identification, as well as post-translational modification studies. So let us first start about basics of MALDI. So MALDI is an efficient process of generating gas phase ions of peptides and proteins for mass spectrometric detection. So MALDI is one of the most widely used ionization technique currently applicable in the proteomics area. So this ionization method was independently developed by two scientists named Koichi Tanaka and Helicam Tanaka. They also received the Nobel Prize for this novel contribution into soft ionization techniques such as MALDI. So let us go through the same some of the points and basic concepts involved in the MALDI time of flight. So we can explain that in two parts. One is MALDI, which is ionization source. Another is time of flight, which is mass analyzer. So let us first talk about MALDI, which stands for matrix assisted laser desorption ionization. So analytes of the proteins of interest are mixed with the matrix, which is usually an aromatic compound. There are various type of matrices available, which we will talk in more details when we come to the sample preparation and matrix selection. So, but just for your several references, we can use some example like 25 dihydroxy benzoic acid. So we can use synapinic. There's one acid called synapinic acid, and therefore there are several other choices. So once you selected a matrix for the experiment, then analyte and matrix can be dissolved in an organic solvent. So after which then it can be placed on the metallic target. As you can see in this slide, the first left section shows you how to place the analyte and matrix together on the sample plate. So now once you have placed the matrix, and the analyte on the target plate. So you can put that in the vacuum chamber and apply high voltage. Now these crystals are targeted with the short laser beams, as you can see in the slide. So then rapid sublimation can convert analyte into the gas phase ions. So now these ions, once generated, they can accelerate away from the target plate through the mass analyzer, which is time of flight. So and they can reach towards the detector. Okay. So that is its merits and demerits. So there are some advantages and disadvantages of using MOLDI as an ionization source. So the sample preparation is very easy. The MOLDI provides high tolerance to salt as compared to the electrospray ionization method. So the MOLDI produces single charge species. Most analytes can accept the single photon. The single charge characters can result in some molecules having large mass to charge values. So therefore, the mold is typically integrated with the time of flight mass. So analyzer can be provide the M by Z range for the large ions. So as well as now these are various methods of using molding. Obviously, it has to be connected with the time of flight. Now there are various demerits of using the system. So there is strong dependence on how to prepare good sample for this analysis. So sample preparation methods here will influence the spectrum generated for these experiments. Okay. So now in the next slide we'll cover the time of flight concepts. So the time of flight mass analyzers, they consist of ion acceleration and focusing optics and a flight tube. As shown in the slide, you have a source where sample ionization is occurring due to the laser beam bombardment. So when ions are moving in the time of flight tube and reaching towards the detector, so now often we can also add the reflector and ion mirror, which can increase the path length. So this time of flight tube is measures the mass to charge ratio of ions based on time it takes for ions to fly in the analyzer and strike to the detector. So now the mass is exponentially proportional to the flight time. So how much it takes to travel in the time of flight tube. So ions of the lower masses can are accelerated to the higher velocities. So now time of flight oftenly outperforms the scanning mass analyzer in its sensitivity and scan speed. Okay. So here the time of flight of a charged ion can be calculated by using the equation shown in this slide. So the time of flight is directly proportional to the square root of mass of ion. So now in this equation, T represents time of flight, M is mass of the ion, Q is charge on ion, and V0 is accelerating potential, and L is the length of flight tube. Okay. So here one more brief concept about time of flight. So in time of flight tube, the ions are accelerated to high kinetic energy, and due to the different velocities, they are separated in a flight tube. As I mentioned earlier, by adding the reflectron or a reflector, the ions can turn around in the reflector that can compensate for minor differences in the kinetic energy. So now if you take an example where you have three ions as shown in the dark blue light here and the red color in the slide. So now you can expect that the small ions, which is the red one, will show the first peak followed by the blue ions, and then the sample preparation and sporting will be done after that. So there is one term called in-gel digestion. So here we'll start preparing the sample for the mass spectrometry in multi-top. Just wait for a minute. Okay. So sample preparation and spotting. So here the in-gel digestion. So the protein sample must be prepared suitably before it can be analyzed by the mass spectrometer. So if you have run a 2D gel, so first of all the purified protein of interest needs to be excised from the gel on which it has been electrophoresis and dissolved in the suitable buffer. So depending upon the application, if you have purified a protein which you can separate that on the gel and cut that band, or if you have a mixture of proteins in 2D gel, you can just excise that particular sport. So sport can be dissolved in a suitable buffer called trypsin, which can be used or added by this mixture in order to carry out digestion of the protein. Then trypsin cleaves the protein at the C terminal of the arginine and lysine residue that is not always universal. If you have a proline present immediately after, and it will hinder that, but overall the protein is digested into smaller fragments of called manageable sizes. So once the protein sample has been digested, all the salt buffer and any detergent must be removed from the sample. So after doing in gel digestion and before proceeding for the mass spectrometry analysis, and between the an efficient steps is to use some filters, or we can call it zip tip, which can be eliminate some of these contaminants and salts. So it offers several advantages such as quick verification, sample enrichment, and ensures that there is no contamination. So there are multiple advantages of using zip tips. So however, it can purify only limited volume of the sample, and also it absorbs some amount of the protein samples, so thereby leading to the losses. So the purified protein sample can be mixed with an aromatic matrix compound, such as example like alpha alpha cyano hydroxycinnamic acid, or you can call cinnapinic acid. So in the presence of the any organic solvent. So the components are mixed thoroughly, and then the solution containing the organic matrix with embedded analyte of interest can be spotted onto a metallic molding. So the samples plate moldy gives you an opportunity to analyze large number of samples in high throughput fashion. So the target plate, the laser energy gets absorbed by the matrix and is transferred to the analyte molecules, which undergo rapid sublimation, resulting in gas phase ions. So the gas phase ions here generated are accelerated and travel through the side tube at different rates. Okay, so the lighter ions move rapidly and reaches the detector first, while the heavier ions migrate slowly. So these ions are resolved and detected on the basis of their mass to charge ratio, and a mass spectrum is generated. So parameters such as geometry design, power supply, quality calibration method sample, and morphology, ion beam velocity, etc. All of these factors affect the accuracy of mass detection. Okay, so let's talk about how to prepare the sample. So we'll discuss these steps in more detail. So the first part will be in gel digestion. So the mass spectrometric identification of target protein greatly depends on the efficacy of in-gel digestion process. So that generates a mixture of peptides from the target protein. So through proteolysis digestion. So these slides give you an overview, since in the last module we discussed about two-dimensional gel electrophoresis. So here it shows that if you have a spot of interest, you can excise that spot from the 2D gel and then subject that to in gel digestion, followed by the mass spectrometry analysis. So in gel digestion is a multi-step procedure which includes spot selection, spot excision, removal of stain, reduction, alkylation, proteolysis, cleavages, as well as peptides extraction. Okay, so multiple steps are involved, and how good your in gel digestion is going to ensure the success of the spectrum generated from mass spectrometry. So now, although this overview shows you the process to start with the 2D gel, but the same can be also applied for even the gel-free proteomic techniques. So if you want to analyze a sample even from the gel free gel-free proteomic based approaches, it is often good idea to separate those protein complex mixture on the SGS page. Okay, slowly bend and then extract the proteins on that platform. The gel indigestion can be performed so that you can simplify the proteome and then you can increase the overall proteome coverage. So similar protocol can be modified and used for the various types of application in the proteomics. Okay, so this was for the day, and good luck for your exams. So you guys can leave the session. Thank you so much.